everybody. My name is Murat Elagoz, and welcome to my thesis defense. The title of my thesis is Identification of Transcripts Encoding Enzymes Putatively Involved in the Synthesis of Anti-Inflammatory Compounds in the Diatom Cylindrotheca CLOSTERIUM. I would like to start to present my work by briefly introducing the key elements about the topic, then move to the steps followed throughout the thesis period, explain and discuss my results, and finally summarize the outcome of the project. So, introduction. Diatoms are photoautotrophic eukaryotes, which constitute the most diverse group of phytoplankton with an estimation of 200,000 different species. The diversity offers an enormous reservoir of new compounds with possible biotechnological applications, which is just started to be exploited within the last few decades. They belong to the class Bacillophyceae in the division Chromophyta. One of the interesting things about diatoms is that they are the most prominent biogenic silica producers in oceans due to the construction of their cell wall from hydrated silica. Currently, three main cellular processes are affected by or involve silica in diatoms, which are cell cycle, silicon transport, and cell wall synthesis. For many diatoms, silicon starvation is known to induce growth arrest and development of lipid droplets. Cylindrotheca CLOSTERIUM is a cosmopolitan pennate diatom, which is characterized by motile and solitary cells with a lanceolate shape. As you can see from this figure, it consists of central nucleus and two chloroplasts with long, thin, flexible ends. Recent studies suggest that it has antioxidant and anti-inflammatory properties. The only study on anti-inflammatory activity of Cylindrotheca species is studied by Lauritano and her co-workers in 2016. The study assessed the capability of inhibition of release of tumor nuclear factor alpha in lipopolysaccharide-stimulated monocytic leukemia cells. The study did not reveal any microalgal metabolite that displayed the observed anti-inflammatory activity. Therefore, further investigations were required. So, for my thesis, I hypothesized that Cylindrotheca CLOSTERIUM have transcripts that are involved, that code for enzymes, which are involved in the synthesis of anti-inflammatory compounds. Therefore, my main aim was to analyze the transcriptome of the species in order to identify these transcripts, which encodes for anti-inflammatory compounds, and other biological, biotechnological applications. It is important to mention that this is the first study to investigate the potential genes which are involved in the synthesis of anti-inflammatory metabolites in diatoms. Furthermore, in order to stress the microalgae and activate the broadest range of metabolic pathway of interest, a silica starvation experiment was performed. Materials and methods. The first step was culturing Cylindrotheca CLOSTERIUM. So the media was prepared by Goulard's F2 medium, and for silica starvation experiment, very minimum concentration of silicic acid was added. 3 replicates of start culture was added to either media with complete Goulard's nutrients or media without silicic acid. Later on, the cultures were incubated at a climate chamber at a specific temperature and light intensity, 12 to 12 light to dark cycle. The cells were harvested when the stationary phase was reached. Afterwards, RNA extraction was performed, and in the meantime, genes were started to be selected. For reference genes, genes that are already used for other microalgae as reference genes were selected. And for genes of interest, the genes that encode for enzymes with biotechnological interest, here anti-inflammatory compounds, and as well as maybe other biological interests were selected. Primer tree software was used to design all the primers. Later on, the RNAs were retrotranscribed into cDNAs, and polymerase chain reaction was performed with the designed primers and template cDNA. Afterwards, gel electrophoresis was performed and bonds were extracted and sent for sequencing, and the raw sequences were analyzed by using BLAST. Two main RT-qPCR plates were prepared, one to determine the primer reaction efficiency, correlation factor, and standard curve for oligonucleotide pairs by serial dilutions of cDNA. Another one as the experimental plate, and 1 to 10 dilution factor was used to be able to allow gene amplification within the optimal read window. In order to assess intra-array variability, all RT-qPCR reactions were performed in triplicates, and no template negative controls were also added. For normalizing the expressions of selected gene of interest, a panel of reference genes were first screened in the experimental conditions, and the identification of best reference genes were carried out by using 3 different softwares, which are GeoNorm, NormFinder, and BestKeeper. The gene expression analysis was performed by REST tool, and statistical analysis was performed by GraphPath-Prim statistical software. Results. During my results, I will only use the abbreviated version of genes because some of the genes names are fairly long. For the reference genes, 6 genes were chosen out of the all the genes that have already used for the other microalgae, which were actin, calmodulin, AFL, GAPDH, alpha and beta tubulin. And the function of the genes are already given here. When it comes to genes of interest, the selection was carried out with the enzymes involved in the synthesis of anti-inflammatory compounds or enzymes of biotechnological interest, and therefore it was decided that galactin-9, MGD, MPGES2, PIJA, PIK3C3, PDDLNS3, and VDE will be used. The first 6 genes were involved in the synthesis of anti-inflammatory compounds, whereas the last one is involved in the breakdown of a specific anti-inflammatory compound. These are the designed primers for the selected genes, and the range of the designed primers were kept between the 19 to 21 nucleotides, and for the length of amplicon, 150 to 250, with the efficiency from 93 to 100. As you can see from this picture, all samples shown visible 28S and 18S ribosomal RNA bonds. Also, the visibility of each bond is actually correlated with the RNA concentration of each sample, and the smaller bonds over here are actually derived from the plant cell organelles, such as chloroplast. When it comes to range of RNA concentrations for the samples that are cultured, we can see that in control conditions, 825 to 1,705 nanograms per microliters was obtained. And for the samples in silica starvation, 954 to 1,524 nanograms per microliters. In terms of purity of samples, rate of absorbance at 260 nanometers and 280 nanometers, as well as 260 nanometers and 230 nanometers was used. For all the values of 260 to 280 were around 2, so that means that the samples were pure. When it comes to 260 to 230, except the samples 2 and 4, they were also around 2. When it comes to these 2 samples, which were around 1.5, they were characterized by the typical common impurity, which is due to the triazole RNA extraction. When it comes to reference genes assessment, in order to determine the best reference genes, the first software we used was the BestKeeper. And as you can see, the lowest standard deviation used was for the tubulin alpha. And when it comes to for the second software, which was GeoNorm, it was actin and EFL with the lowest average expression stability. And last but not least, for the norm finder, JPDH was suggested as the best reference gene. So as you can see, all three of them had different best reference genes during the ranking. Therefore, for the normalization of the data, tubulin alpha, JPDH, actin, and EFL are selected and used. When it comes to genes of interest of expression levels, this figure summarizes them, and basically the control is this line, which is the Y line, and these genes are upregulated and these genes are downregulated. According to REST analysis, only 4 genes of interest involved in the synthesis or inhibition of potential anti-inflammatory compounds showed significant downregulation under silica starvation, which were galectin-9, MGD, PIGA, and VDE. Discussion. This is the first study which focuses on identification of transcripts coding for enzymes that are potentially involved in the synthesis of anti-inflammatory compounds. As we saw from the previous slide, 4 genes were significantly downregulated under silica starvation, which means that the metabolic pathway involved in the synthesis of anti-inflammatory compounds are only active when the species is cultivated in control medium, and inactive when grown in silica starvation. This is parallelly in line with the previous study that was done by Lauretano in 2016. And the best reference genes that was used during this study can be beneficial for the successive molecular studies for this microalgae in these culturing conditions. Furthermore, the future studies can combine the molecular study with the chemical analysis to prove that these are the actual genes that are responsible for the anti-inflammatory activity of this microalgae, as well as genetic engineering could be perceived as another field for the future. To sum up, not only the hypothesis is accepted, but also the aim is achieved. For the first time, by transcriptome analysis and gene expression level analysis of the selected genes of interest, it is shown that in the transcriptome of Cylindrotheca CLOSTERIUM, there are genes that encode for anti-inflammatory compounds. These are the references that I used. I would like to thank my supervisor, Dr. Chiara Lauretano, for her continuous support. Thank you for your attention, and I will be happy to answer if you have any questions.